Hi, folks. This is Dr. Surasinghe. We are moving on to our next topic in population ecology within our general ecology lecture series. This is about, we are going to talk about in this one, population growth and regulation. All right. So let me always get my pointer here. That helps a lot. Okay, so now at this stage, I believe that you guys have already started working on your human demography lab. So the things I'm teaching over here should not be that new or unheard of, right? So these things I expect you guys to know this material relatively well, and also this particular part about life tables, these calculations I'm going to go over could be useful for you to complete your labs, because I assume that you should be different levels with with respect to human demography lab. So what I am going to teach you will be relevant to what you will be doing, particularly when you're doing the data analysis for that particular lab, right? Or if you have already done that data analysis, this will be a very useful revision. So, what is a life table? Let's talk about that first. Life table reflects or is constructed based on birth and deaths of individuals in a given population. Um, and also, based on the life table, you can forecast future population trends because it actually tells you the proportion of individuals are surviving um, in each age class, in each age category, right? So therefore you can understand the future trends of the population based on a life table. The life table provides a summary of how survival rate and reproduction rates change, or how survival rate and reproductive rates differ among different age classes of a particular species. Now, speaking of which, life tables are constructed for a particular population, so it only reflects survival and reproductive success, reproductive rates of a particular species, right? In fact, just one single population, right? We do not construct life tables for different species. So the example life table you are looking at to your right over here, it shows uh, the survivalship or survival of naturally germinating seeds, right? So let's take a look at what's happening over here, right? The first is actually age. First column is age, right? So, and in here, these numbers corresponds to like 3 months time frames, right? So, 0 corresponds to the initial period, right? 1 corresponds to a particular to those grasses that are 3 months in age. 2 corresponds to grasses that are what, 6 months in age, so on and so forth. So a single age category, right, equals a period of 3 months, right? And you have to keep adding it up as you go further. Okay, that's, and then the next column is number alive. So um, number alive corresponds to the actual number of individuals that are alive, that are present in each age class, that completes that particular age class, right? Over here at age 0, at the very beginning of monitoring of this grass population, there were 843 individuals to begin with, right? And uh, when that particular population reaches 3 months, right, in age, you only have about 722 individuals surviving. So what you can see is, as the population ages, the number alive declines. It is very intuitive, right? Grasses are living organisms, they are annual plants, so you would expect fewer plants to survive as the population ages. And also remember that when you're looking at this germinating seeds, all of the seeds came out, were produced at the same time, right? So at the beginning, the entire population um, belongs to the same cohort. Okay, and I will talk about that particular term earlier, population. So third column SX survival rate, that is actually the age specific survival rate, right? Because like each row corresponds to survival rate of individual age classes. It is the probability of an individual class surviving from a particular age class to its subsequent age class, age X to age X plus 1, right? So in other words, if you look at the SX, at the beginning, there were 843 grasses, okay, individual plants, in other words, right? And then um, only 722 of 843 survived to 3-month-old plants, right? And let's do another age class. In this case, um, look at this age class 2, which corresponds to 6 months. Sorry, let's look at let's look at that particular age class, right? Age class 2, which corresponds to the grasses that are about 6 months old, right? So you get about 527 surviving, right? And that started with 722. So if you want to calculate the survival rate for the second age class, that is the age class 1, you have to divide 527 by 722, and then you get 0.73. Okay, so that's actually how you calculate a survival rate, right? The survival rate for a particular age class equals what? How many individuals in that particular age class survives into or moves into the next age class. Again, how many individuals of a particular age class survive into the or move into the next subsequent age class from age X to X plus 1, right? So that's as easy as that. Let's move on. So another way to interpret this is that only 85% of seedlings will reach the age of 3 months, right? And then the next one is 0.73. What does that mean? Only 73% of 3-month-old grasses will reach the age of 6 months, right? And again, you can go for the below, right? It says 0.6. That means only 60% of the grasses will reach the age class, the next age class, which is actually the 9-month-old grasses, right? So far and so on. You can keep interpreting the survival rate. So as we continue on to the other columns, I do also want you to tell you another way to look at a life table. Look at the life table as a numeric representation, a mathematical representation um, of the fate of the population, that is determined, that is calculated, objectively calculated based on that particular population's survival chances and their reproductive success, right? So that's another way of interpreting what a life table is. And well, we will move on to the next column, survivorship, LX, right? So survivorship is the proportion of individuals surviving at a particular age since birth. Right? It is the proportion of individuals of a particular population surviving or remaining alive at a particular age since birth. Now, by default or by rule, the first age class of any population for any life table should be 1. Right? Now, you should have understood by now that the survival rate will always be a fraction, right? It will be always below 1, okay? And then in certain cases, actually, it could be 1, right? So 1 or below 1 is actually what the values of survival rate can take. Now the same rule regarding survivorship, right? It could be 1 or below 1, right? So L0, L naught, or like the first age class's survival is always 1, right? You can write it down whenever you do any calculations. And how about the next age class? Well, how do you calculate for the age class 1, which corresponds in this case 3-month-old plants? Um, you divide 722, the number alive in that particular age class, by the number alive in the previous age class, which was 843. How about plants that were 6 months old? Well, how do you calculate the survivorship for that? Well, what is the number alive for age class 2? Well, that is 527. Well, then divide 527 by the number alive at the beginning, right? That was 843, right? And then you can move on from that particular point onwards, right? And then how about the next age class, which corresponds to grasses that are 9 months old? Well, 316, well, divided by the number alive at the beginning, 843. So when you calculate survivorship, you do see that the denominator is constant, right? Denominator is always going to be the number alive at the beginning of that particular population, or the number of individuals present at the youngest age class, right? Or number of individuals that were present at birth, right? No matter how you interpret it, it's going to be a constant. Right? When you calculate survivorship, denominator is always going to be a constant. Well, how would you interpret that? Um, you can interpret it as, I always wanted to like to get the laser point. Okay. How do you interpret those numbers of survivorship? Well, for the age class of 1, right, for the 3-month-old grasses, the survivorship seems like 0.856, so roughly 0.86. So that means about 86% of the seedlings will survive to become 3-month-old plants, right? And if you go further down, the next survivorship is actually 0.625 for the age class 2, right? What does that mean? About 63% of the seedlings in this population will become 6-month-old plants, right? And then the next or the last column, right? Now remember, some of the life tables may not have the last column, but some of them do, right? It depends on your objective, because in certain cases you are not interested in calculating the fecundity, or calculating the reproductive success. In that case, the last column is not important, right? But in ecology, if you are actually interested in not just studying what's happened in the population in the past, or what is the survivorship, if you want to know more than that, if you want to know like future trends, you need to actually calculate fecundity, which tells you the reproductive success of that particular population. So fx, fecundity, means the stands for the age specific average number of offspring produced per female. Well, in simple English, it's actually the number of number of babies produced or reproduced by females in a particular age class, right? So in this example, if you look at F1, right, for the 0 age class, right? Um, Not F1, for the 0 age class, right? What is the fecundity? 0. What does that mean? Well, meaning that those plants, of course, at birth, right? Those plants that actually, those tiny little seedlings, right? That is coming out of the seed, have 0 fecundity. They cannot produce seeds. In other words, of course, because you are looking at, like a baby plant. And then if you look at F1, which corresponds to 3-month old plants, right? What is their fecundity? 300. So if a 3-month old plant can produce on average 300 seeds, right? For a plant, the babies are seeds, right? I mean, I'm an animal biologist, right? The kind of science I do is more related to animals. So hence I keep calling them babies. But anyways, for a plant, seeds are their babies. And for F2, what is it? 620. What does that mean? A plant that is about 6 months in age can produce on average 620 seeds. As simple as that. Um, so as you can see, what happens to the survivorship as the population ages? Well, clearly it declines as expected, right? Well, in generally, that's actually what you would see, right? And then what happens to this survivorship? Well, of course, that also in this case declines as the population ages, right? Well, what happens to the fecundity? You can also see the fecundity actually, increases first with the aging population, and then it declines as the population continues to age even further. Um, so it's kind of interesting to understand and try to interpret what happens to number alive, survival rate, survivorship, and fecundity as the population ages. Now, there are two different types of life tables. You should already know that since you have started, most of you have started your labs. Um, so one type is cohort life table. What does that mean? Well, cohort life table follows the fate of all newborns of a given population um, from their birth to death, right? So cohort life table pictures or it shows the fate of all newborns of a given population um, until their death, right? So what is a cohort? That's actually another good question that you need to ask. A cohort is actually all individuals that were born at the same time. So a cohort life table um, tracks the fate of a cohort, right? Meaning it tracks the survival of all the individuals that were born at the same time from the time of birth to their death, right? I mean, as you can see in this table, right, the survivorship goes all the way until it is 0. Okay, meaning um, these grasses does not live beyond the age class 8, right? That actually should correspond to 24 months, and in fact grasses don't live more than um, a year, right? So 24 months is actually about 2 years. So that kind of makes sense, why you don't have any individual surviving beyond that point, right? And even if you look at the age class 7, that is what 7 multiplied by 3, that's 21 months, right? You only have 3 surviving. Again, quite sensible calculations, right? Grasses are not supposed to live that long, right? All right. And so can you apply cohort life tables for any biological population? Seems like it does work for grasses. Well, yes, because grasses do not live that long. As you can see, they don't live beyond 2 years, and generally most of the population perishes after a year, right? Um, so it could actually work a lot better for short-lived plants, particularly short-lived grasses or short-lived herbs, right? Um, how about long-lived trees like deciduous trees or pines? Still, it could work. Um, as because plants are very sessile, right? They don't move around. Like when you plant it somewhere, they stay there, right? So yeah, but plants could live longer than you, so it could be a little bit challenging to apply a cohort life table um, for deciduous or coniferous. It's not impossible, right? If you have many people to take collect data, right? If you have like a research lab where one after the other person, inherits the project, you actually can calculate and construct a cohort life table for plants. You can also do the same for sessile animals, right? Animals that do not move around, right? Like coral reefs do not move around. Sponges do not move around, like they anchor themselves and stay there, right? Sionas, those lower codates, so any sessile animal, right, can be followed, at least theoretically speaking, with respect to a cohort life table, right? Because you can track when they are first when they first settle, right? And then you can actually trace their survivorship after that. In fact, this has been very successfully applied for sessile crustaceans like barnacles. Let's move on. And by the way, the calculations and the meaning of this, the formulas for these calculations are already presented at the bottom of the table, as you can see. So other type of life table is called static life table, right? So that is applicable for many animals that are highly mobile, right? Most animals move around; they don't stay in place. So cohort life tables are hard to apply unless you actually put some sort of a radio collar or some sort of a transponder inside that animal, and provided that you have enough manpower to individually track uh, each and every of those animals, you cannot construct a cohort life table for non-sessile animals. That's when static life tables come handy, and it's kind of fun to do static life tables. So those are constructed based on survivorship and reproduction of individuals of different ages during a particular snapshot of time. So if you recall, right, from the previous slide, cohort or age specific life tables, right, also known as dynamic life tables, are constructed based on data collected by following a cohort throughout the cohort's lifetime, right? Now, that kind of an approach is rarely possible with natural populations of animals. Even it is also difficult for particularly for humans, right? And and again, just to remind, a cohort is a group of individuals all born during the same time interval, right? And now, static life table, also known as time specific life table, right, shows age distribution data that are collected from a cross section of a given population at at a specific time frame during a particular period, right? Um, and sometimes that kind of data collection could happen through mortality data, and that's exactly what you guys are doing um, in your human demography lab, right? You're actually going by visiting in a cemetery. You are looking at individuals um, tombstones, and that's actually collecting mortality data, right? And that is not a single cohort, because individuals that you are collecting data from, you collected data for about like what 100 individuals, and you know that they are not all born at the same time. So that's actually the life table you constructed for that lab is not a dynamic life table or a cohort life table. That's a static or a time specific life table. Okay, now, in order to construct that kind of life tables, static life tables, you actually need to visit a particular area like you did in the case of the human demography. You visited the cemetery, and for animal research, you have to visit a particular habitat, right? Probably repeatedly during a certain well-defined time frame, right? Maybe a single season. So in this case, you go to the field, right, and you employ a particular sampling strategy, a sampling technique, right, a trapping technique, or an active survey technique, and capture individuals. Well, then the biggest challenge is how do you determine their age? Well, in your lab, luckily, I mean, you are looking at humans, so you can look at the tombstone, right? You, um, for animals, you have to have some other method to estimate, right, the age of an individual that you quote. So you need to use some sort of other characteristic that reflects their aging, right? So you need to know for that particular animal population you study what kind of biological features are indicators of the age. There are many ways to do this, right? If you look at fish scales, on the top diagram, we are looking at a fish scale. Actually, that is not a fish scale. This is a fish scale. What am I looking at? Yeah, this is a fish scale, right? The If you put a fish scale under a microscope, you are going to see this distinct growth rates, right? Now, those growth rates that you see, right, corresponds to a growth of the fish in a particular given year. In every year the fish survives, every season the fish survives, it actually places an extra growth ring, right? So by counting the growth rings, you can actually estimate the age of that particular fish, right? Now, mind you that you cannot randomly pick any scale and reliably count the growth rings, and you can actually clearly see that in summer the growth is much more pronounced. You can see much more larger rings during summer. During winter, as you can clearly see, the growth is much more shorter. Look at the width, of the winter growth is actually few millimeters. Seems like that, and um, the growth ring for summer is maybe like 100 times the size of the winter growth ring, right? So and then the actual annual growth ring is actually collectively the winter growth and summer growth, right? So if you look at it over here, that's the first year growth, right? And then the second year growth, this particular winter growth plus summer growth, second year growth, like that. That's actually how you count the number of years, right? You have to tally up both seasons. Um, and actually, you might actually ask the question, okay, so where is the fall growth? Well, fall growth could be embedded in the summer growth. And then we have the spring growth. Well, it could also be embedded between winter and summer growth, right? So, but basically, you cannot identify all the four seasons. You can clearly see the summer growth, which actually could contain some of the fall, entire summer, and late spring. The winter growth can contain late, maybe late fall, uh, early spring, and the entire winter. Okay, so larger, understandably larger scales, right, actually are easier to use for age estimation, and smaller scales are difficult, not because they don't have the growth rings, but it is difficult to observe them, right? And of course, if the scale is damaged, it's extremely hard to calculate it, right? So you have to have an intact scale. What are the other things that you can use? When it comes to deer, right, this is actually a deer, and a photograph of the deer lower jaw, right? The tooth wear pattern is also an indicative of the deer's aging, right? When the tooth of or the teeth of deer is actually have, they have a very tall crown, right? So this tooth, the starts when they actually start with, they actually are much taller, right, in the crown height. As the deer ages, it keeps eating, right? As a result, the teeth start wearing off, right? So the teeth get shorter and shorter as the deer ages, right? So by actually calculating the extent to which the, the teeth has wear off, you can estimate its age, right? And that particular graph actually roughly shows how you can kind of estimate the age based on different types of teeth. And in fact, you have to compare apples to apples here, right? If you're looking at um, um, you have to compare like molar to molar, premolar to premolar, right? So you cannot like cross them. So you have to actually look at the same teeth, across different deer that you capture, right? And this can be done particularly when you get even if you encounter like a fresh cure, right? Even if you find a like a skull, right? You can actually reasonably estimate the age at death of that particular individual deer, right? Based on the same technique. What else can we use over here? If you look at this particular diagram, right, you are looking at a turtle shell. On the carapace, you can kind of see in these plates, right? Now these plates are actually modified scales, right? Reptiles also have external scales and fish have internal scales, right? So these distinct lines you can see are actually distinct scale development, right? So what happens when reptiles grow is that they actually produce, any reptile can actually produce newer scales, right? In snakes, they shed their older scales, but turtles keep their older scale, right? The tiniest scale you see on the top, right, is actually the first scale they have when they are born, right? When they actually emerge out of the shell, emerge out of the egg. And as they age, right, in every year they actually produce a new scale, right? So that actually corresponds to 1 year, right? So if you can actually reasonably count these um, layers of scales, you are actually counting their age. Now, can you do it on the plastron? Theoretically, you can, but the problem with the plastron or the bottom shell of a turtle is that that shell um, continuously gets abraded, or is under the friction force as it travels, right? It could rub against the substrate. So some of the older scales or the early scales can get kind of rubbed off and can fell off the plastron, but on the calaparis it does not happen, right? So in the calaparis there's a greater likelihood of retaining older shells than the plastron. So that's actually one way you can actually age turtles. Um, and you can also do the same by look into the bones, right? And in fact, you can do this for lizards and amphibians, right? So you can actually take a small snippet of amphibian or frog fingers or a salamander finger, right, or a finger of a gecko, and put it under a microscope and count the growth rings in their bones, right? For this purpose, you have to actually look at a long bone, like a phalanx, like like a bone from a finger, right? Take a tiny snippet. You don't need to take out the whole limb, right? You you don't need to take out the whole digit. You need it like a tiny little snippet of the finger. Take a section of it, put it under a microscope, right, and look at the cross section of that digit. And if you stain it properly, you can actually easily count the number of growth rings, and each growth ring corresponds to a particular year, right? In that way, you can actually estimate how old a particular um, lizard is, or how old a particular salamander is, or how old a particular frog or a toad is. And this is actually used when you are doing amphibian monitoring or reptile monitoring for endangered populations, where monitoring their age is extremely important. And by the way, that kind of techniques are very less intrusive. You might think about, oh my god, are you taking a bone away from it? No, you are taking a tiny part of it, right? And you do know that most amphibians and certain lizards have a very high regeneration capacity, right? Um, so as long as you get some bone in your snippet, your tissue sample, and as long as that represents a not a flat bone, but kind of a long bone, like a tailbone or a digit bone, right? That's good enough for you to calculate their age very reliably. And of course, like I mentioned, there are certain staining techniques that if you use them properly, if you apply the correct dye, the staining technique, right, you can very easily calculate distinct ages or distinct growth rates and calculate the ages reliably. The top one that is a mystery I saved for, that's actually called otolith. Right, otolith is the middle ear bone that you find in fish. Right. So how do you do this? Is actually you go to a particular habitat, you throw a net, you catch a representative sample, right, of that particular population. Well, the sampling technique varies, right? If you are looking at a lake, it's hard to electrofish in a lake unless you have a very specific boat designing for electrofishing, right? So you have to cast a net, like gill net, or deploy some sort of suspended pike nets and collect that sample, right, and then you have to sacrifice those fish, right, or jab a forceps underneath their operculum, underneath their gills, right, break the bone that surrounds their middle ear, and then you grab the otolith, right? Otolith is a bone, like a middle ear bone, and then you take the otolith out, right? Now, that process sounds very tedious. Well, when you start it, yes, you'll have to have it'll take some time. Like you first might need to even dissect the head, then carefully find the otolith structure, and then you break the otolith covering, and then access um, the actual otolith, right? Because that otolith is actually a very fragile structure, although it's a bone. If you are not careful, you can break it. But with practice, I have seen fisheries biologists, particularly marine fisheries biologists, that that can easily do this, like in matter of 5 seconds. They will grab the fish, jab the forceps, right underneath the operculum, goes behind the gills, right, and kind of break the otolith cover, um, and then take the outlet, right? All in matter of like not more than 10 seconds. Sometimes that's fast as 5 seconds. I have seen people doing that, and I have done this myself, but it takes time for me. And I'm not a marine biologist. I study fish, but I'm not a marine biologist. But it takes time for me. When I first trained myself to do it, actually took quite a while, like something around like 10-15 minutes to access the order link. But with practice, actually, I could do it under a minute, but not within seconds. So what you do after extracting it is actually that's not done. Once you extract it, you have to kind of rub it against a sandpaper to flatten it almost to the level of a scale, right? So when you flatten it, what happens is it becomes thinner, right? And now um, you can put that sanded otolith under a microscope, right? And then you see what I see over here. Now, what the picture you see over here is actually um, taken up from a very high powered microscope, right? And you can actually use a scanning electron microscope to do the same. The scanning electron microscope can scan through the otolith, right? Um, and in that case, if you're using a scanning electron microscope, you probably don't have to sand the otolith, right? Because it scans through and you can clearly see the growth rings in that case. But if you don't have a scanning electron microscope, you have to go with a high powered light microscope. In that case, you will have to sand it so you can actually kind of create a thinner version, a thinner section of the otolith and count the distinct growth rings, right? All the rings you can see over here that I highlight with my diagram corresponds to different years, right? Particularly in the northern temperate zone, it's not that difficult to see distinct growth rings because of the seasonal differences. Um, there are clear enhanced growth during summer and the opposite during winter, so you can clearly see the growth rings. But this, as in a tree, um, you cannot easily do the same thing for tropical organisms, right? In a tropical tree, if you take a cross section, you cannot clearly see the growth rings clearly, okay? Um, but in a temperate tree, you can clearly see the growth rings. The same thing regarding fish, okay? Growth rings are much more distinct in temperate or polar organisms, and hard to see the same in tropical organisms. Now, fecundity, well, I did explain what fecundity means. It's the number of offspring sired by different individuals at a given age, right? So, how do you calculate fecundity with respect to a static life table? Mm, well, what you have to do is actually for that again, you have to go through some sort of a cyclifaction, right? Unless you can actually calculate successfully, observe an individual in the field. Right? For certain mammals, you can do that, right? For large mammals, you can trace a particular mammal, observe it in the field, and see how many individuals it sire, right? Like carnivores, the number of individuals sired is relatively small, right? Particularly for cats, large cats sire no more than like three individuals, right? For bears, like two, somewhere between two to four, right? For elephants, it's actually one for a particular herd, right? So in that particular case, you can directly observe that. But for other vertebrates, right, like fish, amphibians, doing that is pretty hard. Um, so in that case, what you have to do is you have to sacrifice an individual, right? Better if you can identify a female by external features, right? Dissect it and di- take the ovaries out and actually define some sort of a mechanism to count the number of eggs in that ovary, right? And if you have done for this for fish, I have done this for fish for research purposes. It's not easy to calculate the number of eggs in an ovary, right? Because there are thousands, right? In an amphibian, it could be hundreds. So what you do is actually you weigh the entire ovary intact, and sometimes there's a pair of ovary. So you take one of them, weigh it, right, and then take a section of that ovary, weigh that particular small part again, and then um, count the number of eggs in that tiny little part of the section of the ovary, right? And then extrapolate by multiplying it, right? First, for that particular ovary, let's say your entire ovary weighs like 10 grams and your tiny section weighs 1 gram. In the tiny 1 gram section, you have um, 10 eggs. So you multiply 10 by 10 to calculate for the entire ovary because your section is 1 gram, your entire ovary is 10 grams. And then you have two ovaries, you multiply that by 2 to calculate for the entire organism's fecundity, right? So that's a general example of how to do that. Um, I have also read people doing this based on volume by calculating the volume of the ovary, and then but that's harder to calculate the volume of an ovary by displacement method. So I'm not going to go into detail. The weight-based extrapolation method is actually relatively easy and easy to do. But the downside is, as you can see, you have to sacrifice a number of individuals to do this successfully, right? But science comes in. Science comes with some sort of sacrifice, and I have done that, but I for quite a long time. But I have never seen um, the population being severely impacted because you sacrifice a relatively few number of individuals. And also, what you need to understand is actually age, like you saw in the previous plant life table. Age is a very important determinant of the fecundity and survival, right? There's a certain age in a given population where the survival is maximum and the fecundity is also the maximum, right? So in fact, here's a kind of a really interesting hypothetical situation. Okay, if we are looking at a particular population, right, you might actually have the same population size, right, and you could be looking at the same species, right, two different populations. So you might even have the same age distribution, right? Um, but the population that actually have the greatest number of individuals in the most fecund age class will actually grow faster than the other population, right? So think about that. I'm looking at two populations, the same population sizes, right? And let me tell you this, they might have the same average growth rate, right? Um, but they might also have the same average cross-age fecundity. But right, the population that actually have the largest number of individuals for the most fecund age class will grow faster than the other. Because it can grow faster, it can actually easily overcome certain age specific mortality. They might even show greater resilience to mortality, right? They can actually recover from losses faster. So this is really important for conservation applications, right? So when you're protecting a particular population, particularly if you're trying to protect highly exploited populations like marine fish, right? Or a particular carnivore population that is actually suffering from hunting, right? Or some sort of a mammalian population that is suffering from hunting, right? You need to focus on increasing survival of the individuals that have the greatest fecundity, right? The age class that is most responsible for population growth is the age class that shows the greatest fecundity, that can sire the greatest number of individuals, right? And needless to say, right, the age class that have a greater number of females, right, will also disproportionately drive um, the population growth. Okay, let's talk about what you do with life tables. Well, you know the answer by now with respect to your lab experience, right? You construct this survivorship curve, right? So data from the life tables can be graphically represented as survivorship curves. You know, if you look at the survivorship, you know, on the x-axis, you are looking at the age. You know, that is the first column in your life table, right? And the y-axis is the proportion of surviving, um, proportion of individuals surviving um, in the population you study, right? So that is actually kind of the LX, right? This the column before fecundity, right? So there are three types of general survivorship, right? Type 1, 2, 3, right? Now, these three survivorship curves are identified based on the age class at which the greatest mortality versus survival is observed. Let's understand the each type, and that will explain what I just mentioned earlier, right? Type 1, right? This particular type is when, right, newborns or juveniles or younger adults have a relatively greater survival and the death rate increases at very older ages. Okay, so look at this graph. At early age classes, right, you see a very high level of survival, right, and it precipitously declines as the population ages. Um, Examples are like what? Tortoises are actually a good example, right? Um, so tortoises actually have very great survival. Now this is really interesting. Not turtles, tortoises, meaning like what? Those chelonians that live on land, right? Land tortoises, like giant tortoises in particularly, you know, as juveniles, as young adults, they have a very high survival, right? They actually live centuries, right? Certain turtles are known to survive up to age of 400, but after that, they actually their chance of survival declines um, dramatically, right? And you can also argue that modern human population also follows type 1, right? Because of the advances in medical sciences, healthcare, and medicine, um, the baby survival of the prenatal deaths are very low. Okay, newborn deaths are very low, right? Um, right, and juvenile deaths are very low, right? Preteen deaths are very low. Teen deaths are very low, and prepubescent deaths are very low. So they survive up to very older age. You know, currently the global human population ranks around 70s in terms of life expectancy, and after that it actually declines, right? Um, and if you look at developed countries, the actual developed country, the human population of developed countries are a much better example of type 1 survivorship. Certain underdeveloped or third world countries like African nations may actually not represent the typical type 1 because of increased mortality observed among newborns or neonatals. Um, Type 2, right, is actually when, as you can see in this one, where the survival rate or mortality rate remains a constant regardless of the age, right? Uh, Coral reefs are an example for that, and so are birds, right? Generally, because of the parental care and the smaller number of eggs produced, um, survivorship of the young, right, is actually fairly constant, right? Egg hatching rate is fairly constant because of the incubation and the parental care. And post hatching parental care also progresses among birds until the birds actually fledge, right? When the birds fledge, actually they are still able to fend for themselves. And sometimes post fledgling parental care, right? And there are many behavioral mechanisms that actually increases um, survival of younger birds. So the bird survivorship actually remains fairly constant until a particular age, right, where they actually all die. So coral reefs too, right? their survivorship is a constant regardless of their age. Um, Type 3, you observe a much greater mortality and much reduced survival at younger age classes in neonates. You experience a larger, greater mortality and lower survival, right? And once a particular age is reached at older individuals, relatively older individuals, the survivorship increases and the mortality rate declines, right? So you can see kind of plateauing at older age classes, right? So kind of type 3 is the polar opposite of type 1. Um, Most amphibians, freshwater turtles, marine turtles, most invertebrates, right, particularly mussels, follow the type 1, type 3 survivorship. Okay, moving on. Um, so we're looking at population pyramids of two countries. On the left, you see Nigeria. On the right, you see Japan, right? My question is, depending on the contents of the graph, which country will have a higher population growth rate? Now, answer this question not by your general knowledge, but by what you understand about population growth, survivorship, and reproduction, right? Compare the two graphs for a second, right? Their population sizes by 2010 are not that different. Of course, Nigeria has slightly high, 558, millions, and in Japan it is 127 millions. Well, that's not huge. That's not a huge difference, right? Well, the difference is fairly lower, in the scale of millions. And if you look at the life expectancy, right, seems like the maximum life expectancy for Nigeria is around 85, right? Japanese population, you do see, as by your general knowledge, they actually live a bit longer. So there are some individuals surviving above the age of 85, but generally in terms of the average life expectancy, those two countries are comparable, right? It's kind of interesting because Nigerian medical and health services are pretty good, have high standards. Probably that's why. Um, and but if you look at the actual population pyramids, the distribution of individuals within each and every age classes are not the same, right? So what is an age class? Age class represents the member of a particular population whose ages fall within a specific range, right? So you can clearly see that the Nigerian human population has this typical pyramid shape. There's a whole lot of individuals in younger age classes, right? And fewer individuals in old age classes, right? And if you look at Japan, right, the number of individuals in different age classes are not that different. Of course, the younger and middle age classes slightly have a greater number of individuals, right? But more or less, on average, right, the number of individuals in each age category are comparably similar, right? Um, so, despite comparable overall population sizes, despite the life expectancies, maximum life expectancy being comparable, right? Which population will grow faster in this case? Well, the population that has a greater representation of younger age classes that have the greatest fertility, that has the greatest fecundity, will actually grow faster, right? A. So if you have a higher representation of young age classes, will grow faster. B. Those actually have a greater representation of old age classes will grow slower. The reason is actually younger individuals have a greater fertility, right? And actually they can actually successfully deliver babies, right? They will have fewer birth complications compared to older individuals, right? They will suffer less abnormalities during the delivery process. They will suffer less abnormalities in their gamete production, right? The gamete viability in younger individuals are higher, right? And we also know in terms of humans, right? Um, ovulation ceases in female after older ages, right? All right, that's a really cool example to highlight my point regarding how important it is, the age structured distribution, um, to predict what happens to the future in that particular population. So in fact, it's estimated through population modeling, mathematical modeling, it is estimated by 2050, um, Nigerians will have a population size about or upwards of 300 million. In contrast, it's expected the Japan population to actually shrink slightly downwards of 100, like, 100 millions. So the population that actually today has a comparable population sizes will actually end up with dramatic different population sizes because of the differential growths, um, coming from the differences in the age specific population distributions, right? It's a really interesting example. Right, moving on. So what are the pragmatic values of life tables, right? Why do we construct them, right? They are mathematically burdening. They're kind of fun, at least in my point of view. Like, well, why do we do that? So life tables, survivorship curves represents age specific mortality rate or risk of death, right? Because of that, we can actually understand which age groups in a given population are more susceptible to death, right? Is it newborns, young adults, or the actual adults or older individuals, right? We can actually determine that. That is important in wildlife management, in conservation, as well as in the insurance trade. I mean, if let me talk about the insurance trade first here, because it's kind of more applicable to us or to you. For me, wildlife is more applicable. Um, so in the life insurance trade, they actually do use these kind of ecological principles, right? They do construct life tables, and they construct survivorship curves for different populations, because when they issue you a policy, they need to do a risk assessment, right? What is the risk of you dying, right? Particularly if this is a life insurance, right? Your benefactor will actually inherit a large sum of money, right? So, so because of that, life insurance agents, right, or the policymakers, they need to calculate, when a particular individual signs up for a life insurance, what is his risk of mortality, right? Given his health conditions, given his current age, given his demography, given his ethnicity, given a wide variety of factors, what kind of population does he represent? In that particular population that he's representative of, that you are representative of, what is your risk of mortality now and in future? Based on that, they actually calculate your premium, right? So if you have a greater risk of mortality, they are going to give you a huge um, premium, right? You will have to pay a lot, right? But if you are actually much younger, and you represent a particular population that is very low in mortality, then you will actually continue to age, and your life insurance policy will accumulate more money, so you will pay a smaller premium. Okay. Similar kind of things happens in health insurance as well, right? It's actually not susceptibility to death, it's rather the susceptibility to an illness, right? That's also very, population related parameter, where you can do exactly the same. Instead of survivorship, it calculates your risk of healthiness or losing your good health. In terms of wildlife management, how does this come? So if you are managing an endangered population, right, of a plant or an animal, you need to understand first, what age classes are more susceptible, right, to mortality, right? um, for example, let's look at turtles, right? Many freshwater turtles, and almost all marine turtles, are endangered or threatened with human activities, right? In order to protect them, we need to understand, like, naturally, um, what kind of factors increase their mortality. If you look at marine turtles, their mortality risk is the greatest in young age classes, for many reasons. One, naturally, most of the turtles that emerge from do not emerge from the nest, right? They die immediately after they hatch. And as young hatchlings move from their nests into the sea, they can get predated. So there's an increased predation risk as young. If you look at human predation, right, human collection overwhelmingly targets young turtles, eggs, right? People collect eggs, right? It's really horrible. And in fact, because of all of those risks for turtle conservation biologists, particularly I actually do work on turtle conservation in southeastern Massachusetts. I actually collaborate with SUNY New England to protect one of the endangered species in the country, Blanding's turtle. Um, so what we do is we actually collect um, nests, right? Eggs from the nests. As immediately as they hatch, we actually collect them, right? And we take them into an aquarium and grow them up to the age class of 9, right? So roughly up to 9 to 10 months, we grow them ex situ outside their natural habitat. The idea is to feed them, take care of them properly, and then keep them in aquarium conditions that are healthier, that are disease free, that provides the optimal temperature, optimal light conditions, and provide with optimal food. And when they're about 9 months old, why we release them after 9 months is because after that their survival in the wild is relatively high. So with that, we can actually we boost their population. So what do we see? We pick the part of the survivorship curve where they are most susceptible to death. We remove that individuals from the natural population, boost their chance of survival, reduce their mortality risk, and put them back into the wild when they are past that threshold age, right? Um, so that's actually one example, right? And there are many examples you can think about. Uh, so we can, with this strategy, we can construct different life tables and construct different survivorship curves, so different populations of the same species, right? And understand differences in their survival and reproduction. And in that way, you can actually understand what different environmental or external variables are responsible for different patterns of survivalship and mortality, right? If you look at this, my example right up here, you're looking at conifer survival, right, in the Pacific Northwest, California, and interior, right? And if you look at the graph, you can see the interior populations have a relatively lower mortality, right? And the Californian populations are faring much better. Pacific Northwest are intermediate, right? But the interior populations are doing pretty bad. So when you want to plan conservation strategies, right, to protect coniferous forests or softwood forests, right, you can actually focus conservation in the interior populations first. Provide more funding to those. Invest more research dollars to understand what is happening on these populations. And also, if you look at the top image, you're looking at the same species, human populations. We are comparing US female survivorship versus Gambian populations. And we are actually splitting the Gambian population into two cohorts. One um, born during the kind of the hungry season, which is actually represents the drought seasons, and the one that are actually surviving that were born afterwards. And we can clearly see that there's a distinct survivalship differences between the US populations and the Gambian population. Right? Even within Gambia, we can clearly see a difference between those that were born between July and October, the so-called hungry season, are actually faring less well compared to those born between November and July outside the hungry season, right? So Gambians born between November and June lives much longer than the Gambians born during the hungry season. The reason is, like it's kind of interesting too scientifically, because those who were born during hungry season actually don't live that longer, and they actually still suffer increased mortality, right? Even after they age, because the initial investment that they received from their parents in terms of parental care, initial growth are much lower. So they're actually less healthy. So those less healthy babies, is unlikely um, to catch up to their equivalents that were born much later, so up to other cohorts that were even born during the same years. Really interesting to see how impactful the early age criteria is. Like it seems like it actually there is a sort of a legacy effect, sort of a carryover effect. If you are not born under ideal conditions, right? Um, the biological impacts will persist much longer, right? It's also interesting if you look at the survivorship in early age classes up to the age of 15 between the two Gambian cohorts are not that different, right? They almost the survivorship curve almost superimposes on each other. The survivorship differences actually catches up after teenage age, right? Beyond 20 years or older, you can actually clearly see that the differences in survivorship magnifies, right? So it's kind of an interesting observation. So what that tells you is actually by understanding constructing survivorship curves, you can understand, you can calculate population growth rates and project what will happen in future, right? So you can understand population trends. Now that's not only important in the human populations, like in the Gambian population, but it is also important in conservation of species. It's important in timberland management, right? If you're managing a particular timberland, you need to increase the survival of trees until they live up to the harvesting age, right? So when you manage a particular timberland, you need to understand and project the future population trends and future survival of harvestable trees, right? And in that way, you can actually, when you know that, you can manage your timberland in a favorable way to increase your ultimate timber yield. Um, and this is also really important if you are particularly in the timberlands. This is extremely important. One of the biggest problem when you are managing a timberland, meaning you're managing a forest for harvesting trees for timber or paper or pulp, you need to protect your timberland from pest infestation. There are lots of forest pests that will completely destroy your timberland, right? So if you are controlling a particular pest, right? Let's say hemlock woolly adolgid, which creates problems for eastern hemlock, right? The best way to get rid of that pest is by understanding the pest's life cycle, right? In their life cycle, what life history stage is mostly susceptible to natural deaths? If you aim your pest management strategies to that particular age that suffers the greatest natural survival, right? That has the lowest natural survival and highest mortality, right? If you focus that particular age class, you will be successful at eliminating that particular pest, right? Um, and also, it's also important to understand, right, in terms of pest management, right, what is the age of oviposition of a particular female pest, right? And if you can steer, focus, direct pest management strategies on the females of the highest fecundity age class, you will be also successful at controlling that pest population. The same thing if you're trying to get rid of a particular parasite, right, from a particular wildlife population, right? Again, if you focus uh, at the parasite's most susceptible stage of the life cycle and deliver some sort of a medicine to counter at that particular life history stage, you will be successful at salvaging your host population, right? The same thing regarding getting rid of invasive species, right? For conservation biology, getting rid of invasive species is extremely important and extremely hard, right? So if you identify for your invasive species the most susceptible point, if you recognize the Achilles heel of your invasive species by pinpointing towards the most susceptible age class, you will be successful. If you pick um, and also if you pick the females of that invasive species. And if you understand when does those females reach their greatest fecundity and target those females in terms of invasive species management or eradication strategies, you will be more successful in getting rid of that particular invasive species. All right, folks, with that actually I am going to finish the first part of the lecture on populations, and I will see you very soon with the second part of the same topic.